Bismillahirrahmanirrahim. This is the second video. So this is the second video on the June 2022 paper 4-1. I have done question 1, 2 and 3, and hopefully in this video we'll do 4, 5, 6 and 7. As you all know, time 2 hours, 100 marks, and you have to do this paper in time, and you have to be very careful how you handle this paper. Question 4. In 1973, a technique for genetic engineering was used for the first time. Recombinant DNA was made using a plasmid, and this was successfully transferred into an organism. In 2012, a new technique for genetic engineering called gene editing was developed. Table 4.1 lists some statements about the two genetic engineering techniques. Complete Table 4.1 to compare the original genetic engineering technique using a plasmid vector with the newer technique of gene editing. For each row, place a tick in the correct column if the statement applies and leave a blank if the statement does not apply. So either you know it or you don't know it. So if you don't know it, please don't waste your time and trying to do it. It can produce a transgenic organism. Gene engineering using plasmid, yes, it can. Gene editing, yes, it can. So you got 1 mark for that. Then it can modify the characteristics of an organism. Yes, genetic engineering, you put the insulin into the bacteria and it will modify, it will start making insulin. So yes, and gene editing will also modify it and it will produce a protein or whatever in which we've done it. Then the third one is it can delete unwanted DNA. It can delete unwanted. Genetic engineering using a plasmid can't do that. So, but gene editing can result in deletion. So we leave this blank. Then it uses an enzyme that cuts DNA. Yes, we do it in both. It uses an enzyme that cuts DNA. Of course, we use it in both of them. Then the last one, it can use RNA to precisely locate the target gene. Well, not in this one, but yes, in this one it can. So as I said, either you know this or you don't know this. So these 5 marks are for those students who know it, or you can just guess it, but either you know it or you don't know it at all. So I always say genetic engineering is the last chapter that we do, and sometimes some students find it very difficult to be able to do it. And I said, well, just leave the question on it. If you feel it's difficult, you're wasting your time doing it and you are not comprehending it. So we will remove this. So this is how the table goes. Orange trees, Citrus sinensis, produce fruits that are important food crop. The functional leaf area of orange trees may be reduced by the growth of citrus canker bacteria. These bacteria cause citrus canker disease. Scientists use gene editing to develop 2 types of orange trees with different mutations. The mutant orange tree leaves showed resistance to citrus canker disease. So you see, citrus is important fruit. Functional leaf can be reduced with this bacterial disease, so the leaves will be less photosynthesis, less crop yield. Now what they did is they changed the DNA and that resulted in resistance to the citrus to the disease. Forget the name of the disease. Figure 4.1 shows the area of leaf with citrus canker disease in wild type, not gene edited. So the wild type is the original one, which is not gene edited. And then the gene edited original leaves after they've been exposed to the bacteria which causes the disease. Now you see this table here. So type of orange tree A, B, C, area of the leaf with disease was 2.2. So we start writing this here 2.2 and here it was 0.4. Always do that, always write it. And here it was nearly 0.2. So A had type of orange tree A had 2.2 millimeter square area which had the disease. The B had only 0.4 millimeter square area which had the disease, and C only had 0.2 millimeter square area which had the disease. So first explain the question to yourself and then read the whole question and then read it again and then start solving it. Identify the letter that represents the wild type of orange trees. Now, how would you guess that? Because it told you something in the question. What did it tell you in the question? Figure 4.1 shows the leaf areas in wild type and they've been exposed to the citrus canker. And then it says the mutant orange showed resistance. So the one which showed resistance means that they won't suffer from the disease. So naturally, it would be the A which is suffering from the disease must be the wild type. So identify the letter. This would be the A, because as I told you that the mutant ones were not having the disease. So this is something which they expected you to understand from the question. If you read the question carefully, only then you could have done this. Otherwise, if you didn't read it carefully and just sort of did it very casually, which is a very good habit of all of you to do things casually, then it says explain the social benefits of this example of gene editing. Oh, very easy. Social benefits, 3 marks. What were the 3 marks? More, if we use the mutant trees, then they will have more food, improves the quality of oranges, more income for the farmers, and cheaper oranges, and spend less on bactericidal chemicals or pesticides, which is going to kill the bacteria which was causing the disease. Now, just writing it, I know is easy. Increasing the yield would, of course, mean more fruit available because the leaves would not be spoiled by the bacteria. So, improves the quality of oranges. They'll be large sizes. They'll be more juicy because now there's more photosynthesis because the leaves are not being destroyed by the bacteria. More income for the farmers, and then fruit will cost less. And we won't have to spray chemicals to kill the bacteria. So, less pollution of the environment, chemicals or pesticides which we use. Question 5. Two subspecies of reindeer, whatever, live in North America. Members of the different subspecies belong to the same species but have some morphological differences and are found in different geographical locations. Figure 5.1 shows a reindeer. Please remember, you must do this paper before you look at this video. So you must try to solve it beforehand and do it in writing so that you know where your mistakes are. If you just look at my video, you're not going to learn much. Figure table 5.1 compares the features of the North American reindeer species subspecies. So we have the habitat that was south woodland warmer, north tundra colder, type of food trees, leaves and grass. Here there was lichens and moss. Summer and winter feeding grounds overlap, yes, no. Carry out long migrations, no, yes. Body size large, small, and color of fur dark and light. Please read this very carefully and then read the rest of the question. And coming to the A part of the question, during the last ice age, an ice sheet separated southern and northern populations of R. tenandus in North America. Explain how this ice sheet affected the evolution of R. tenandus to result in the two different subspecies. Now, if you know your syllabus, this is what? Speciation. So it is what? Geographical isolation. Or if you said allopatric speciation, when they are, I tell you this, alag, separate. So allopatric speciation, so there's no interbreeding, there's no gene flow between the populations. Now in both the places, there are going to be different selection pressures on each population, weather, grass, food, light. So different mutations in each population, then adaptation to the environment or the habitat or the food or the vegetation, and then that results in different gives different morphological and behavioral differences. Naturally, you know that you've studied in selection and evolution how the habitat is going to affect how there are going to be selection pressures. What animals will benefit if they are larger seeds or smaller seeds, what sort of beak sizes they have. So all that you've done that before. So you just need to, if you know any question on selection and evolution, is just these mark scheme points. So, I mean, just wording it allopatric speciation, or if you said geographical isolation, you got a mark, then no gene flow between population because now they are separated by this ice sheet. Then there's different selection pressures. Each area has different weather conditions. Then adaptations in that environment, in that habitat, they adapt to that environment. This gives them different morphological and behavioral differences, and that is because of could be different mutations which occur. So each population will have a different set of mutations because of their environmental pressures. Then we come on to the B part of the question. Assess the relative importance of natural selection and genetic drift in producing the different colors of fur of the two subspecies of reindeer, and number two, the different body sizes of the two subspecies of reindeer. First, let's look at the definitions of natural selection and genetic drift. Natural selection is traits that improve survival or reproduction accumulate in the population, and adaptive change, survival and reproduction. Genetic drift is what? Frequency of traits changes in a population due to chance events, random change. So a chance event results in the frequency of traits which change in a population due to chance events. So you see this, you see the person putting the foot and killing a large number of these beetles. Now, assessing the relative importance of natural selection and genetic drift in producing the different colors of the fur of the two subspecies. Now, the color of the fur is natural selection. Why? Because the darker the fur, which is more adapted in the woods or in the warm or in the shade, and they're probably better camouflage. So, right colors have protected them against predators. But in this situation, the different body sizes of the two species of reindeer. Now, this is could be genetic drift. You could have also said not natural selection. Why? Because the small size in the north was beneficial. The small lose the heat, harder to keep warm, smaller fat reserves, not good with unstable food supply. Less food in the north reduces the growth or the size. So this is what you have to understand is that you see one was natural selection and one was not a natural selection. You could have said genetic drift as well, but you could have said that as well. Now, different body sizes is dependent on what? Was dependent on the environment because of the weather and because of the food available and all that. So please remember, whenever this question comes, you need to be very careful in selecting your answers and writing your answers. If you look at these answers, there's usually very general knowledge. You see, now, like here it says natural selection, dark color selected for good in woods and camouflage against predators. And here it says genetic drift, but you could have also said not natural selection. And the small size in the north is not beneficial. And the small lose more heat, so they heat faster. And then less food in the north, so less growth. So you have to really pick up all the points from the question and come to general conclusions about this whole story. Part C of the question, hybridization has occurred between individuals of the two subspecies, which now live in the area previously covered by the ice sheet. Comment on how the hybrid populations compare to the pure subspecies in terms of genetic variation and potential to adapt to climate change. Now, they're telling you in the question that the hybridization has occurred and now they live in the area previously covered by the ice sheet. So that means the hybrids are a better bet. So the hybrid populations have more genetic variation or more alleles. They have alleles from both the subspecies. They have more potential to adapt. Then the alleles for migration may let some find new habitats. The alleles for warm and cold or for different temperatures are present in them. That is why they can survive in this area which was previously covered by the ice sheet. So hybrid, more genetic variation or more alleles, and alleles from both the subspecies are present in the hybrids and can adapt better in the future. Then they might have the alleles for migration, and this may let them to find new habitats. And they have alleles for different temperatures, so that is why they have survived. And coming to the last part of the question, D, outline how practical techniques could be used to test the hypothesis that migratory behavior in reindeer has a genetic basis. So whenever they ask you such a question, it's a very simple thing. How do you study their genome? So you take some blood or you take some tissue or something to get their DNA, and you test both the subspecies, the migratory and the non-migratory, and then you use the PCR to amplify the DNA, and then you use gel electrophoresis and compare the results, or you see the genetic differences. Then you sequence the DNA and identify the differences or count the differences or the changes. Then you check for correlation between the DNA, genetic profile, the alleles, and the migratory behavior. Does it tally with that? And then you use database or bioinformatics to find the role of the gene sequences that you have figured out. So basically, in any of these, you had to get the DNA because what did it say? Genetic basis, practical techniques. So, in practical techniques, what do? You take the DNA, you amplify the DNA, you do gel electrophoresis and all that. This is something which you just have to remember, and you can write this in any question. Now just going through it, the wordings of it: obtain blood or obtain DNA, test both the subspecies, use PCR to amplify the DNA, use gel electrophoresis to see the genetic differences, sequence the DNA and identify the differences, test for a correlation between the DNA and the migratory behavior. Is there some correlation? Do the migratory birds have some DNA which is common to all of them? And then you use the database and the bioinformatics to really actually figure it out that which genes are being expressed here. What are the alleles? Is there any different alleles that we thought would only be present in the migratory and those not present in the non-migratory ones? Question number 6. A part: in mammals, the blood glucose concentration must be maintained within narrow limits so that the body cells can function efficiently. Name the mechanism by which the blood glucose concentration is maintained within narrow limits. What? That's very simple. Homeostasis. Now, this is something which I'm sure everybody knows, and you couldn't be making a mistake in this. You could have said homeostasis, you could have said negative feedback, but I mean, of course, Cambridge allows negative feedback. As far as I'm concerned, I won't allow that. But I'm not the Cambridge examiner, so it's fine. You all can get away with it. Glucagon is released by the alpha cells of the pancreas when the blood glucose concentration decreases below the set point. Figure 6.1 outlines the response of liver cells to glucagon, right? So you've got glucagon receptor, G protein, enzyme A, cell surface membrane, ATP, second messenger B, activated kinase enzyme, enzyme cascade, and activated glycogen phosphorylase. Now it says state how glucagon reaches the liver cells. Very simple, through the blood. How will it reach? Through the bloodstream, or you could have said the blood, or you could have said the circulation. So you could have said either the blood or you could have said circulation. Then it says with reference to figure 6 point, name enzyme A, which is adenyl cyclase. Adenyl cyclase. Now that means they expect you to know the names of these. There's something which you need to remember. Adenyl cyclase. And then name enzyme B and second messenger B. What is that? That's cyclic AMP. Cyclic AMP. Or you could have said chota C and AMP. Not the capital C, capital letter, small letter C and AMP. So cyclic AMP. So that got you your 2 marks for that. Then coming to the part 3 of the question, state the role of the enzyme cascade. What is it? That amplifies the signal, or you could say expands the message. So amplifies the signals. Enzyme cascade basically amplifies the signal, makes it, more and more other and more proteins or enzymes are activated. Then it says state the function of the final enzyme in the pathway glycogen phosphorylase. What does it do? It breaks down glycogen, because why would glycogen be broken down? Because the blood glucose level is falling. That's when glucagon is released. So it will break down glycogen, and more glucose will enter the bloodstream. So break down glycogen. That's only when you're fasting, and that is the time these days when just before iftari, lots of glucagon will be released because you have been fasting since a couple of hours. So break down glycogen, or you could have even said glycogen to glucose. That's fine. You just could have put an arrow glycogen, arrow glucose. And then coming to the C part, a biosensor is used to measure blood glucose concentration to detect that it is within the normal range. Describe how a glucose biosensor works. Now that's very simple. That's a very direct question. How does a biosensor work? It works very simple. Blood on a strip or a pad, and then you have on that you have glucose oxidase. So the glucose is converted to gluconic acid and hydrogen peroxide, and this is an electric current or a flow of electrons, and this there's a proportional, but however much the glucose is in the blood. So it's 50 glucose and it's proportional to the concentration, and this will result in a numerical or a digital reading. So the electron flow is proportional to the glucose in the blood. So if a person has 100 glucose, so 100 electrons will flow, and that will result in a digital readout. Similarly, more glucose, more electric current will flow. So more current, more. So these are the things which biomedical engineers are going to figure out how to develop these machines, and that is what I tell you is the field of today in which you need to study instead of becoming a MBBS or something. That's what you need to do, biomedical engineering. So blood on the pad or the strip which is inserted into the glucometer or the biosensor, and that contains glucose oxidase. The glucose oxidase catalyzes a reaction. The glucose is converted to gluconic acid and hydrogen peroxide. So if it's 100 glucose, 100 gluconic acid and hydrogen peroxide, 200 glucose, more of the hydrogen peroxide. This generates an electrical current. The current is proportional to the glucose concentration, and then it gives you a digital readout. So there were about 6 mark scheme points, but any four of these would get you the 4 out of 4. Now coming to question number 7. The fruit fly Drosophila melanogaster usually has red eyes. Oh, good, nice, red eyes. A gene for eye color has 4 alleles: red, apricot, honey, and white. Wow, interesting. Define the term gene and allele. Now this is simple. If you don't know this, I feel you should drown in the Ravi River. So gene is the length of DNA which codes for a polypeptide. But can you add up on that? It actually codes also for a gene is for a polypeptide or a nucleic acid. So where does the tRNA come from? Where does the rRNA come from? That also comes from the DNA. But of course, that's not for A level. It's a little higher than A level. So that's why I talk about it. They might ask you a question on that. So what is gene? A length of DNA coding for a polypeptide. Simple. What is an allele? Alternative form of a gene. So these are things we've been doing in O levels, so everybody should know how to write it. So length of DNA coding for a polypeptide or a nucleic acid. I haven't written that, but I want you all to remember that. And allele is an alternative form of a gene. Now, looking at the B part of the question, the allele for red eye CR is dominant to the other 3 alleles. So that means all the red will be CR, CR will be red as well. CRCA will be read as well, CRCH will be read as well, and CRCW will always be read. Why? Because it said the allele for red CR is dominant to the other 3 alleles. So you must do this exercise. You must do this exercise in understanding that it is dominant to the other 3 alleles. So this is something which I just did with you. Then it says the allele for apricot eyes is dominant to the honey eyes. So the apricot is always going to be either going to be, what is the apricot going to be? They're going to be either CACA or they can be CA and CH because it says dominant to the allele for honey eyes. So the apricot ones will be either CACA or CACH. And then it says the allele for white eye CW is recessive to the other 3 alleles. So the apricot can also be CACW. So CW is the recessive allele, and the ones which are white eyes will always be CW. That is it, can't be anything else. Why? Because it has to be homozygous recessive, because it says the allele for white is recessive to the other 3 alleles. So anything with CW is going to show the recessive trait. If it's in a heterozygous state, it is going to be, so CRCW will be red, CACW will be apricot, CHCW will be honey, and CWCW will be the white eyes because that is recessive to the other 3 alleles. Now it says construct a genetic diagram to show the genotypes and phenotypes and the ratio of the offspring. So you have to give me the genotype, the phenotype, and the ratio of the offsprings from a cross between a fruit fly with red eyes CRCH and a fruit fly with apricot CACW. So they've given you this, they've given you the parental phenotype and the parental genotype. Don't like that. I don't think that's a very interesting question. So you are only going to get 3 marks for it. So it's not a very nice genetics question. I like difficult genetics questions. And then you just have to complete it. So the gametes, you just write them separately. So CR, CH, and CA and CW. Just write them in a slide, separated, little apart from each other, and this got you 1 mark. Then offspring offspring genotype. So this will be CRCA. CRCA, will be red eyes. And then the other one will be CR and CW. and this will be again be red eyes, right? And then we have CHCA. What will this be? And what did it say in the question? CHCA is dominant to the allele, so this will be apricot, apricot. And then we have the last one would be CHCW. We know that W is recessive to all of them, so CH will be honey eyes. So this is how we would have done this question. Now the ratio is what? The ratio is the red are 2, and apricot is 1, and honey is 1. So 2 ratio 1 ratio 1 would get you your 3 out of 3. You see, this is 2 reds, 1 apricot, and 1 honey. So it is 2 ratio 1 ratio 1. Now it says describe how you would carry out a test cross. Now, how do you carry out a test cross? You always cross it with a known homozygous recessive. Now, which one? So cross with the white one. And the white is what? CWCW. Because this is the homozygous recessive. Now you know that the red, because we had to find out it says describe how it carry a test cross used to determine the genotype of a red-eyed. Now the red-eyed can be only CRCR, CRCA, CRCH, and CRCW. Now we want to find out what is the genotype of the red. So when you cross it with CWCW, then all the ones, if all are red, then it's CRCR. If some are red and some apricot, then CRCA. And if some are red and some honey, then CRCH. And if some are red and some white, then CRCW. Well, simple. If all red-eyed, then CRCR. Why are we saying that? Because CRCR into CW will be all offsprings will be CRCW, and these will be all red. So if all red, then CRCR. If some apricot, then CRCA, because they would have the A in it, and then they combine with this. You see, you're crossing it with a known homozygous recessive. And if some honey, then CRCH, and if some white, then CRCW. And I've done that for you, the last one, to make you understand this. What do we mean cross with a known homozygous recessive? And this is why how it will tell you the genotype of the red-eyed. Because the red eyes could have been any of those options. We finish this video here, and we will continue in a third video the remaining questions. And thank you once again, and please leave your comments, and if there's anything else I can do for the A2 syllabus, please do let me know.